from the University of Turku.
Thank you very much. If you were to look at this photo of me with my grandmother and think, there's nothing particularly unusual about this, you'd be absolutely right. And yet, in the grand biological scheme of things, there is something incredibly unusual. An individual, here my grandmother, who is in an extended period of post-reproductive life. Why is this unusual, though? Well, life history theory predicts that reproduction and lifespan should end more or less at the same time to maximize fitness. And yet humans are one of 5 species, along with these toothed whales, to show an extended period of post-reproductive lifespan, physiological post-reproductive lifespan. Females of these species live long after reproductive cessation, so understanding what may have led to this extreme decoupling of somatic and reproductive senescence could help us understand what makes us human. One of the main adaptive hypotheses for the existence of post-reproductive life, or an extended post-reproductive life, is the grandmother hypothesis. Very simply, the fitness benefits from helping your grandchildren select for longevity. And there's a wealth of empirical and theoretical support for this, not as the sole driver, but as a contributor in the evolution of post-reproductive life. And there are 2 pathways one can gain fitness as a grandmother, either by increasing the number of grandchildren you have, or by increasing the number of grandchildren who survive to maturity, and can therefore pass on the genes further still. However, context dependence of grandmothering is unknown. When we think of other cooperatively breeding species, be they birds or smaller mammals, we know that whether help occurs at a certain instance and the outcomes of said help on the offspring can vary greatly by context. With the grandmother hypothesis, however, we don't really have any clue about what contexts grandmothers are actually beneficial or not. And this is needed to know the role it actually played in the evolution of post-reproductive life. In this figure here, we can see the fertility rates of women. So we've got age along the x and fertility rate along the y-axis. The lighter blue on the left is just the fertility rate of mothers. So you have a peak in your 30s, and by the time you get to 50, menopause, no more kids. On the right-hand side, we have effectively the grandmother fertility rate. And so we see a similar pattern. It starts to increase, reaches a peak, and then declines. However, in the past, lifespan was limited to the mid-70s, more or less. And yet we can still see that grandchildren are gained at the far end. The opportunity is still existing beyond lifespan. There is still quite a high fertility rate, even as a grandmother gets to 90, or even if she's dead, when she would have been 90. And yet, if we look at the hazard of death, we see it increases. So this basically tells us that the likelihood of a woman dying in any given year is increasing with age. And effectively, we can read this as a kind of increasing hazard, increasing senescence rates. So there's this opportunity for grandmothers to still help, except empirically we see that they're not living long enough. So if grandmothers have selected for post-reproductive lifespan, why couldn't it have selected for a slightly longer one? And do the benefits to survival of grandchildren change as the opportunity to help declines? To do this, we looked at grandmother age effects on grandchild survival to age 5, which in pre-industrial populations is when the vast majority of childhood mortality would have occurred by. So the population we're using here is pre-industrial Finland, 18th and 19th centuries, and we have a wealth of data from church books. These contain information on births, marriages, deaths, and so on, and from these we can reconstruct the genealogies that allow us to assess whether the grandmothers would have had an effect on survival or not. People generally lived patrilocally, which means they were living with their paternal grandmother. But the maternal grandmothers were still living close by. So first I'll talk you through how to interpret the next figure, and then go on to explain the results. So here we have just a figure showing the odds ratios. So this, in effect, is telling us the likelihood of a certain condition either improving or whatever compared to the baseline. So we have a baseline of 1. So effectively, there is no effect of the grandmother. And if you're to the right of it, and your confidence intervals are clear away from the line to the right, then that's an improvement in survival. There's an increased probability of you surviving until the age of 5 with their grandmother present. And if it's to the left, it's a decrease. So with maternal grandmothers, we have the age classes on the y-axis. We see that there is no effect of grandmothers under the age of 50. Now, this could be potentially to do with reproductive conflict hypothesis. And this is basically saying that in our evolutionary past as humans, there was some kind of conflict between mothers-in-law and daughters-in-law for reproduction, and the daughters-in-law won it. But I don't really have time to go into this, so if you want to know more, and it's also been seen in killer whales, if you want to know more, read those papers as well as others. We then see, for the age class of 50 to 75, what could be considered typical grandmother age. We get a confirmation of grandmothering benefits. But then grandmothers 75 and older, so sort of towards the end of the lifespan, there's no late life benefit. It overlaps with 1. The situation is slightly different, however, with paternal grandmothers. Again, we see no effect of grandmothers under the age of 50 on grandchild survival. But we also see no effect in these typical grandmother ages. And we actually see that older paternal grandmothers are detrimental to the survival of their grandchildren. In other words, it was better to have no living paternal grandmother than it was to have a living one who was old. What might this be? Well, it's not killer grandmothers, although that would have been much better sell to the media. Instead, we think it's some kind of resource competition. As I explained before, paternal grandmothers were typically co-resident. So it's possible that there was some kind of resource competition between the dependent generations. As the grandmother gets more and more frail, the parents are having to split their time between their dependent child and the grandmother. And because the maternal grandmothers were close but not under the same roof, that's why maybe there was no effect seen anymore. They're no longer providing help, but because they're not under the same roof, they're not demanding resources constantly. And so we wanted to look into this a little bit more. Is there a cost of co-residence? And so here we have, along the y, another one which is showing odds ratios, but along the y-axis we have the years to grandmother death. Now, this was a pre-industrial population, as I mentioned before, which meant it was pre-healthcare. So if you were very frail and ill, you weren't going to last for very long. There was no medicine that was going to save you. So we were using the number of years until the grandmother died effectively as a proxy of their frailty. And what we found was that the weakest paternal grandmothers, those a year from death, those who were likely to be most frail, were detrimental, again, to the survival of their grandchildren. However, any further away, more years than that, we did not see any effect whatsoever. So this suggests that there's some kind of cost to co-residence, and this is something we're now looking into in the future as well, building on this work. But what does this all mean for lifespan? Well, grandmothering might have had a role, because we saw it, but it can't select for lifespan indefinitely. We do actually see a similar-ish pattern in other non-industrial populations. So this is from hunter-gatherers. We have age along the X and the number of kilocalories along the Y. And for women who are in their reproductive years, they're consuming more than they produce. They're looking after the children, they're having more children, and so on. But when they get into their post-reproductive period, they're then able to help more with the foraging. Their production they are then net producers. But then when they reach their 70s, once again, their ability to forage is declining, and so they become net consumers again. So we see this pattern, so not just in agricultural populations like in pre-industrial Finland, but also this increasing frailty, almost, we see it in all sorts of societies across the world. And so basically, grandmothers are good, but they're not always good. They're not good forever. So just to conclude, grandmothers can be beneficial for grandchild survival, but older and weaker, and more than likely co-resident grandmothers, detrimental. And that grandmothering could have selected for lifespan, but only up to a point. And with that, I'd like to thank my co-authors and supervisors, these other people, and you all for listening. Thank you very much.
We have time for questions. There's one here in the front.
I suppose there would be selection to stay fit and healthy as a grandmother as long as you could, but then once you reach that frailty stage where you became detrimental, you should experience mortality as rapidly as possible to get yourself out of that resource competition situation. Is there any evidence for that?
We haven't really looked into that, and the difficulty with this question as well is or what we were looking at as well is we can't really, you can't say one led to the other. Did grandmothering lead exactly to this, or because it was longer, did grandmothering kind of pull up to that point? Yeah, it's a difficult one. I don't think there's an easy answer, and with the nature of the data, you're dealing with stuff where it's happened in the past. All we're dealing with is records and getting what we can out of those.
I guess you could look even at present day health data.
Can you please use the microphone?
Suddenly,
Could you repeat the question?
I guess you could look even at present-day health data and see whether humans tend to die more suddenly when they reach a certain point of old age compared to other closely related species like chimps or something like that. I don't know.
Well, it's slightly different because chimpanzees don't have any kind of post-reproductive or an extended post-reproductive phase of life. And with contemporary populations, it's quite difficult because we don't really see, because of healthcare, we don't have these incredibly high childhood mortality rates, so we don't need grandmothers as much. So the role of grandmothers has actually changed greatly from pre-industrial era until the present day. Now grandmothers help with either schooling, they can help with encouraging breastfeeding, so nutrition, mental health benefits, some physical health, financial support, things like that. So grandmothers are hugely important still, but not as much so in an evolutionary way.
Hello, I have a question about uncertainty of being actually a grandmother in case of paternal grandmothers, because probably there's a difference between maternal grandmothers and paternal grandmothers, since the paternal grandmother doesn't have to be actually the grandmother.
Yeah, that's normally a big issue for historical populations. But there was work done in 2016 by Martin Lamassoure showing that the rates of extra-pair paternity were actually much lower than estimated. Plus, with our population, there are various reasons why that wouldn't have been the case. And if you're living with your paternal grandmother or your mother-in-law, you're maybe less likely to cheat if you're in the same place. Thank you very much.
Let's thank Simon again. Okay, our next speaker is Robert Lynch from the University of Turkey.
Hi, thank you. So I'm going to present to you the results of a study on the reproductive outcomes of a population of young girls who volunteered or were members of probably the largest paramilitary organization in history in Finland in World War II. This is a paper that's in revision right now. So life history strategies are about trade-offs. You have fast life histories, shorter lifespan, earlier reproduction. A mouse is an example. Slower life histories, longer lifespans, and later reproduction. An elephant would be an example of that. This is true across species. It's true across 48 species of mammals in that graph. And it's also true across countries in humans. This has been known for a long time. The relationships are quite high. So Tanzania, shorter lifespan, earlier reproduction. And Finland, for example, longer lifespan and later reproduction. It's also true of shorter lifespans tend to go with just more offspring in general. And longer lifespans go with fewer offspring. This is total fertility on the y-axis and life expectancy on the left. Okay, I thought this slide was, we can talk about this later. This is a experimental evidence of this using college undergraduates where people who'd grown up poor and were given a New York Times article, like a mock-up of a New York Times article saying that mortality rates in the future were going to go up. And then they asked them about their reproductive strategies. How many children do you want? When do you want to get married? Educational trajectories. And so it's unique in that it's experimental evidence on this, but it's a pretty specific population. It's considered a weird population, Western, educated, industrialized group of undergraduates. This is one of the first empirical studies. It's one of like a classic in human behavioral ecology by Daly and Wilson, where they actually took it down not from the nation to within cities to actually within neighborhoods. And they showed that the top 10 Chicago neighborhoods with the highest homicide rates, women had earlier median age at births. And the neighborhoods with the lowest homicide rates, the opposite was true. This study's been cited over 1,000 times, or close to 1,000 times. It's 1997, and it's one of the watershed papers on this topic in humans. So some of the mechanisms that have been hypothesized to affect reproductive timing. Life history theory predicts that organisms in unstable environments will accelerate reproduction. And some of the most plausible triggers are experiencing stress and trauma during childhood usually, or just observing higher rates of mortality, like if none of your grandparents are alive or your brother died, you just notice it, and that becomes part of your, it triggers some sort of adaptive response. The psychological effect is presumed to involve discounting, which is just how we value current versus future payoffs. And higher mortality rates, lower expected lifespan should increase discounting rates, all else equal, at least theoretically. And the effects are expected to be strongest for younger pre-reproductive individuals. Okay, fine. As you probably can notice, there's a lot of problems with this type of research. It's hard to study, especially in humans. There's this huge problem of socioeconomic status. Obviously, Finland and Afghanistan have a lot more going on than age at first birth and lifespan. There's cultural differences, there are poverty differences, there are educational differences, there are technological differences. The list is almost endless. There are cultural differences that are often not accounted. Even within Chicago neighborhoods, there are different cultures going on. Father absence is a big issue. Father absence is known to lower the age of menarche and decrease the age of first reproduction amongst women. That's often not controlled for. There are family effects. There are different effects of large families and siblings. Parental investment, of course, is a big. There's genetic effects. In summary, there's an endless variety of known and unknown shared demographic variables within comparison groups. So our question was, what effect does exposure to higher mortality rates in childhood have on the reproductive outcomes of women? Our predictions were girls who are exposed to higher mortality rates in childhood will wait less time to give birth. They'll have accelerated reproductive schedules, which just means shorter interbirth intervals. And overall, we might expect that they just would have more children. So I came here to Finland. We had a population of evacuees from Karelia, and it provided this pretty unique opportunity to study this question. There's a natural experiment. We have a Karelian population, which is the same ethnic group from eastern Finland, the same population of people. They were all evacuated during World War II. We have almost all of them. About 10% of them volunteered, 9% of them didn't. You can think of the non-volunteers as a semi-control group. So during that period of World War II, they were separated. And then critically, they rejoined after the war until we get their complete life histories in 1970. So they were part of the same group, split temporarily, and then rejoined. So volunteers, just to talk about Volunteers for Lottosvard briefly, it was a paramilitary organization, approximately 12% of the female population. Some of the tasks were anti-aircraft, food services, recovery of the dead. But critically, there were 8 to 16-year-old girls who volunteered. They were called Lotta girls. Our study population is about 87,000 women, 9,000, which is about the same proportion, who were claimed they were members of Lottosvard organization. We have a bunch of information on them. Okay, so here are the results. These are from Bayesian generalized linear mixed models. These are posterior distributions. The Lotus volunteers have shorter times to reproduction. That's the bottom blue graph. These are 95% credibility intervals. And then non-Lotas are on top, and it's only true up to the age of about 20. Same is true for younger volunteers, have shorter birth intervals after the war ends. And the same is true, but to a lesser extent, of overall reproduction. Fine. But what about some of the bigger problems with these studies? What about family effects? So we can do better than that. And we had the opportunity to actually look at these women because we have genealogies that we can tie to these women. We can actually look at individual families and we can compare sisters. One sister who volunteers versus another sister who does not volunteer. And then they go back into the same families and back into the same ethnic group and population. So these are these results. The error bars are bigger because it's a much smaller. It's about 10% of the group we can do this for, where you had one sister volunteer and one sister who did not. Younger sisters who volunteer do wait less time to reproduce than sisters who do not volunteer. There's the Lotas, the non-Lotas, and credibility intervals are the shaded lines. And it's somewhat true for overall reproduction, but it's true more of the older Lotas that actually have higher reproduction, the non-Lotas, sorry. Sorry, let me go back to that, just if you want to look at it. So that's that, Lotas versus non-Lotas, the volunteers versus the non. So the conclusions are women who were members of this paramilitary organization who presumably experienced either higher stress or higher mortality, exposure to higher mortality. It's hard to tease those two apart. Wait less time to reproduce after the war ends. They have shorter interbirth intervals and have more children after the war ends in 1945. Then there are non-LOTA peers for the big group, and there's non-LOTA sisters for the more limited sample. These results add to accumulating. There's a bunch of evidence since the Wilson and Daly study in '97 that I would take an entire seminar to discuss, but these results add to that. But as we know, correlation is not causation, which is sometimes a bit overlooked, and we're controlling for a lot of things that they couldn't before, but certainly there's a lot that we still don't know, and we definitely don't know the precise mechanisms involved, which is still in the future. So anyways, thank you very much. These are some of my collaborators.
Time for questions. There's one there, the left.
Hi, really interesting talk. I know you couldn't possibly test for this, but I'm just wondering about personality. And could it be that the volunteers were more outgoing, more individuals as opposed to the non-volunteers, which may then affect their future reproduction?
Yeah, that's right. There are things like that, yeah. In fact, we have another paper on this same group showing that the ones who joined were more likely to have brothers. They're more likely to have husbands who served in the military. So we're arguing that that's sort of like kin psychology. If your kin, you perceive your kin are threatened, you may be more likely to join. And we're getting at some of that with the sisters, but we don't have identical twins randomly assigned. That would be obviously the best. But we're getting a little closer, yeah. But you're right, yeah, personality would be there may be some types, and those types of women are more likely to reproduce earlier, at earlier ages, yeah.
Any more questions? There's one here.
Thank you. What do you think about the following hypothesis, that the major factor is not the stress from seeing such events as well, but I think such young ladies had more opportunity to contact with young men, young soldiers, and this is also, this could contribute to, of course, sexual contact and through that early reproduction.
Yeah, no, that's a good point, but I think it's actually more likely that the opposite is true, is because the women who didn't join would have already had husbands or relationships. So I think that you could argue it either way, in other words. Yeah, there's no reason to believe one or the other, but yeah, it's possible.
Okay.
Thanks, Robert.
from the University of Trondheim.
Hi. So my name is Christophe Coste, and I will present a few results relating to the antagonistic pleiotropy and the slow-fast continuum. And the idea is to try to see what those two theories yield to when you look at them together, and to understand if there are issues and how we can solve it from studying them. So these are pictures that were stolen from just the previous presentation. That's my slow-fast continuum, and those are Williams and Hamilton, who studied the antagonistic pleiotropy. So what's the antagonistic pleiotropy riddle? So first, what's the antagonistic pleiotropy theory of senescence? It just predicts that a mutation that positively affects fitness at early life. This is Williams' own chart in his 1957 paper. But at the cost of late life fitness, may invade the population because it's going to have a net effect on fitness. As Hamilton would put it, that means that selection gradients decrease with age. And now what's the slow-fast continuum? So we just saw that. I'm going to be very brief on that. It's basically an axis of life history strategies that differentiates fast organism with high fertility but low survival, and slow organism with high survival and lower fertility. And those are the seminal paper that described this life history axis. And now we want to study both of them together. So we start again from Hamilton, and we built a matrix, so a Leslie matrix, because this has been studied for populations structured by age or stage, but a chronological characteristic. So this is just a Leslie matrix by age. And to simplify, we use same fertility rate for all ages and same survival rates for all ages. It's a very simple matrix with only two parameters and only one, the first line and the subdiagonal. Moreover, we make sure that the asymptotic growth rate of my population, my Malthusian fitness, is 1. And that's what we get when we want to study the fertility selection gradient. So that's the derivative of the fitness by age. The importance of fertility at all ages, that's for an organism that can live up to 30 years, decreases with age. This is exactly or similar to this line by Williams, but Williams considered non-constant fertility rates. So that's one thing. Now, what's happening here is that below a line that depends on the size of the population, for which selection is weaker than drift, then some deleterious mutations might fix, and they will generate senescence. What Williams says is that you can also have impact of a mutation that have effects earlier in life, when you have a positive effect at an early age and a negative effect at a later age, because of the slope between those. And such mutation that has positive effect here, negative effect here, might invade the population. So now what happens if we look at different matrices with the same properties than those, constant fertility, constant survival, and constant Malthusian fitness of 1, then we can see that they are spread over an axis here. And that's our continuum, slow-fast continuum. So slow organism and fast organism will have very different selection gradients and very different selection gradient steepness. Fast organism has much higher steepness in the population. And now, if we look at this in the context of the antagonistic pleiotropy theory, then it means that above the same line that we had before, above this line, individual that are above this line, so above, will become faster, ever faster. And below this line, they can remain slow because selection is weaker than drift. And so according to both theories, if you look at them together, then we're going to have a dichotomy in evolutionary fates, where slow organisms remain slow and fast organisms become ever faster. And that's not what we see in the data. That's not what we see in nature, because in nature, and according to thousands of papers, there is a slow-fast continuum, hence the name. So in order to try to solve this, briefly, we decided to implement more characteristics than just age in our model. And most importantly, we wanted to implement in an evolutionary demography model cost of reproduction. So why cost of reproduction? First, because they are a fundamental trade-off and a fundamental element of life history theory, but also because when you decompose cost of reproduction into the origin of the variance of the trade-off, then you can see that the genetic variance corresponds to the slow-fast continuum, whilst the individual stochasticity, the variance that comes from at the individual level, corresponds to individual costs, which are for individual on the same position of the slow-fast continuum, the different stochastic realization of the allocation mechanism. So in order to implement those traits, we need to have more traits than age. We need to add one trait that implements individual costs, and that would be parity, the number of births ever had. And in our very simple theoretical model, we decide that these births have a linear impact on vital rates. The more offspring you have, the lower your vital rates. And then we have strategy, which is the axis along the slow-fast continuum, with some individuals or some lineages with high fertility, low survival, and lineages with higher survival and lower fertility. So, and then in order to understand and analyze those matrices that are multi-trait matrices, age and parity, for instance. So this is an age parity matrix. That's age 1, 2, 3, age 1, 2, 3, but this is parity 0 and parity 1. And when you add parity 1, your fertility is lower than when you add parity 0. If you want to, in order to understand the cost of reproduction, we need to understand the impact of the trait that supports the cost of reproduction, which is parity. And to do this, we use trait level analysis, which works by folding a matrix, over one of its traits. So here it's a matrix with two characteristics, age and parity. We fold it over parity, it becomes a matrix over age. It's a Leslie matrix. Those two matrices have the same asymptotic growth rate and the same stable state abundance, but they have but for other evolutionary demography measures, for instance, a key measure reproductive value and sensitivities and elasticities, they have differences, and those differences will enable us to measure the importance of cost of reproduction for this particular organism. So we do that for three different measures. The selection gradients at maturity that we saw earlier. So that's on the slow-fast continuum, and we look at the zero parity fertility rate, so that's the F of the matrix, because S then is like a consequence of it because my growth rate is 1. So on the slow-fast continuum, I can see that because of the individual cost of reproduction, my selection gradients are smaller and less, and the slope is less steep, especially for fast organisms, when cost of reproduction are implemented. I also want to look at the variance in reproductive success, and I can see that the variance in reproductive success is also much altered for slow organisms compared to fast organisms. And now I want to combine those, because we know that the variance in reproductive success has a strong impact on effective size, it's completely related, and so on the force of selection. And we want to look at a measure that we call effective selection gradients, which is the selection gradients weighted by the effective size of the population, that to account for both effects. And that's the first graph that we saw, and that's if we look at the effective selection gradient. And we can see that we go from a very steep curve to a much flatter curve. So individual cost of reproduction, they flatten the gradients across the slow-fast continuum. So our riddle, the enigma, came from these differences. Why is it so big? We see that when we implement the most important, maybe the most important parameter of life history, which is trade-offs, then this enigma starts to disappear. So in order to conclude, we can say that the antagonistic pleiotropy riddle, which is why are there so many differences and why we would expect two different evolutionary fates between slow and fast organisms. It comes from two shortcomings of deterministic models that most people use, which is when they are structured by age only, is the fact that trade-offs are not accounting for, and that the effect of individual stochasticity are not incorporated, and they should be. When we apply the trait level analysis on this type of data, we see that this differential is much reduced, but there's still a difference. So if you want to interpret this difference, it might be because we did not implement environmental stochasticity, and maybe genetic costs are a stronger buffer of environmental stochasticity for fast organisms. They need the steepness of selection gradients, while slow organisms don't need it that much because they have individual costs to buffer environmental stochasticity. On another layer, we can see that it's key to implement individual stochasticity into models. Deterministic models don't allow us to understand these things, even for large population. So it's not only a small population issue. And that age might be the strongest determinants of vital rates, but it's most of the time not enough to understand and study life cycles. You need other characteristics as well. And finally, we can see that modeling tools, like for instance multi-trait matrices, trait level analysis, and other tools based on IPMs, matrices, and other continuous time models, can also be used to, they work very well on data, but they can also be used to study theoretical life history questions. So I want to thank you, and I want to thank my collaborators, Samuel Pavard, and the CBD lab, which I'm part of now. Thanks.
Questions? Anybody questions? We have 1 minute. Well, if not, let's thank Christophe again. Thanks a lot. Okay, our next speaker is Nedim Tuzun from the University of Leuven.
Yes, Hello, everyone. Welcome. I'm Nedim from the University of Leuven, as introduced, and I want to talk about the work done in our lab that I think fits nicely within the scope of this session. So, talking about life history evolution, we often refer to intraspecific patterns in any given life history trait, and these are often driven by some kind of environmental conditions. For example, there is this fish species, populations for high latitudes, they actually grow faster compared to populations of the same species from lower latitudes. And that's simply because the temperature up high north is colder, so they have a shorter growing season, so they need to grow fast. That's the reason. Now jumping to another key central element for life history evolution, these are trade-offs. And these are often thought to be driven by some changes in resource allocations. For example, if you invest a lot in somatic growth early in life, you might not have enough energy left later on to perform optimally. And that's exactly what happened for this fish that I just gave an example. The population at the high latitude were found to be more vulnerable to predation simply because they had a lower swimming performance. So there we have the trade-off. Now another famous trade-off, fast growth, short lifespan. That's also thought to be driven by some kind of changes in the resource allocations. And here we want to ask or want to see how the oxidative stress physiology can be part of this trade-off. So talking about oxidative stress physiology, I'm going to quickly summarize the main components. It's going to be extremely simplified since it's a difficult, complicated system. So organisms accumulate over their times reactive oxygen species, also known as ROS or ROS. And to counter that, the antioxidant system comes into play and produces basically antioxidants. They fight these ROS and then try to keep the balance between oxidative stress and antioxidant defense. So what can happen is that you might have too much ROS where the antioxidant defense cannot cope with that. Then comes the oxidative damage into play, which can damage, as I said, some key components of the organism. Then there is this interesting scenario where the opposite happens, where the antioxidant system is actually overcompensating, so producing more than enough antioxidants, which can surprisingly be beneficial for the organism. So this falls within the framework of the hormetic response. When a mild stress is experienced by the animal, it can actually be a positive outcome for the animal. All right, so we want to see how oxidative stress can fit, or the components of oxidative stress can fit into this trade-off cascade, which I depict here. Starting from a fast growth rate, often also comes with faster metabolism, which then produces, which then leads to accumulation of ROS in the animal. And then that often leads to oxidative damage, especially when you don't have enough energy left due to the fast growth. You don't have energy left for the antioxidant defense. That even causes a higher oxidative damage. And all of this might result in a shorter lifespan. So for this experiment, we work with the damselfly model system. That's our pet animal in our lab. So we have a good background information on some key life history evolutionary patterns in this animal, including growth rates. And importantly for us and good for us that so lifespan is kind of a good proxy for fitness for these animals. Specifically, we work with the damselfly Ischnura elegans. Here you see the aquatic larvae, and here two adults in their mating position. So we worked, so we designed the common garden rearing experiments in the lab. And basically we used two different kind of populations, one from the high latitudes, in this case Swedish, and one from low latitudes from France. So the ones at the high latitude in Sweden, the species at this region can only produce a generation every second year, whereas the animals, the species in France, the same species in France, can squeeze in up to 3 generations per year. So that's why they grow faster. So I know this is kind of counterintuitive to the example that I gave with the fish in the beginning, but keep in mind these insects, or at least the life history of these insects, is strongly driven by the volcanism. So they have a fixed time of the year where they can grow, and they grow faster in France to squeeze in the 3 generations. So then the larvae are kept in the incubators in individual cups, the emerging adults in the cages, and then we study the adult and also the larvae. Since I mentioned that we are looking at the role of oxidative stress, we measured some of these components. I'm going to go very quickly over this. So mainly two antioxidant enzymes, the superoxide dismutase, SOD, and catalase, CAT. So these are basically fighting the ROS buildup. We also want to know how much oxidative damage these animals are experiencing. So we looked at the lipid peroxidation of these animals. And finally, we looked at how long the adults live in our experiment, and that is then the lifespan. All right, so I will start with the results. But before that, I will briefly introduce the legend, which will be the same for the following figures. With closed circles, I'm showing the low latitude French animals, with open circles, the high latitude Swedish animals. We looked at growth rates at several different stages of these animals. It's not important for this presentation, but it was consistently higher in French, the growth rate, which was expected, and we already knew that from previous experiments. So now we look at how long the adults live, and then we found that the French animals, which grew faster as larvae, lived longer as adults, and on average 4 days, which is quite crucial. And that is already surprising because that already breaks the fast growth, short lifespan trade-off. So then we look at oxidative stress physiology. Maybe we can get some insight in that. Here I present the first two, in these first two plots, I present the antioxidant enzymes, the SOD and catalase, and how much they were expressed in these animals. And for both of these enzymes, the French animals from the lower latitudes, they had higher levels of these antioxidant enzymes, kind of meaning that they had a stronger antioxidant defense. Now looking at how much oxidative damage these animals were experiencing, the French ones had kind of a lower, slightly lower oxidative damage. So the animals so the French ones had a higher antioxidant capacity, and they also had lower oxidative damage. Kind of makes sense. So to quickly summarize what we found here, the French animals were found to be growing faster, which we kind of already expected and knew. But surprisingly, they also had a stronger antioxidant defense, less oxidative damage, and a longer lifespan. So kind of several components in the flow of the trade-off are not really met here. But we think this all kind of fits into the hormetic response framework, where basically the fast-growing French animals are constantly exposed to a mild stressor, and that accumulates more ROS. And to overcome that, the antioxidant defense comes into play. And in our case, we think that the antioxidant defense even overcompensated, so it produced more antioxidants than actually needed. And we think that actually was helpful for the animal to live longer. So in other terms, the fast-growing French animals had a higher level of ROS during a prolonged time, during the whole larval stage, and that basically increased their baseline antioxidant levels. That's at least our thoughts. But then you might say that all of this is too correlative and at the group level, but we also tried to look with a subset, because not every animal was measured as the larvae and as the adults, because you sacrifice some of them for the oxidative stress physiology. So this first model here is basically a summary of what I showed before. So the antioxidant levels were higher in French animals, so the latitude explains a lot of this variation. When we include how fast they grew into this model as a covariate, the significance of the latitude disappeared, and now we have faster growing animals producing more antioxidant levels. So this kind of tells us that differences in growth rate at least contributed to the latitudinal differences in the antioxidant levels. You might say that you only play with natural variation in growth rate by basically comparing two latitudes. We also tried to, we actually did, but I'm not presenting these results, it's going to take too long, by applying a starvation treatment to basically manipulate the growth rate, so then we have more of a cause-effect relationship. And this kind of treatments are often applied, as you can see, for different taxa by different people. They also looked at oxidative damage and how this translates to fitness. And the results from this kind of manipulation of the growth rates were very similar to the patterns that we found for the natural variation in growth rate. That gives us also kind of a confidence that the cause-effect relationship is also kind of there. And then you might also say, so are these French animals growing fast, more oxidative, more like antioxidant defense and living longer? Are they really superior to the Swedish ones? Well, definitely not, because from our lab and also from different groups, we know that the French animals compared to the Swedish ones have a lower investment in immune function and also lower energy storage. So there might be some costs. You might also say that maybe the cost of this fast growth is not visible in the lifespan, but maybe later on in the life, in terms of, for example, the reproductive output. I definitely agree. And this is what we are currently, or in the next few experiments, we want to try to tackle this, to look at even the second generation. Maybe there comes the cost. So that was basically it, what I want to present. I quickly want to squeeze in that we have 2 PhD positions available in our lab. We're now looking at some microbiome. One position is for the Daphnia magna, how it affects the heat tolerance, and one for the pace of life and how it affects the damselflies. So that was it. Thanks for listening.
Questions? There's one right here.
Thank you. That was very interesting. I just wondered about the body size in these two groups. I may have missed it, but is there a difference in body size and the timing of maturation?
Yeah, so I didn't talk about that. Indeed, it's very crucial. So the faster growth rate was basically driven by the faster development rate. So they really grew faster. They came from egg to the final instar so much faster than the Swedish ones, but they kind of similarly emerged into the same size. So the body size was kind of not different, but it was only driven by the faster development rates.
Any more questions? There's one here.
Hey, I was wondering if you know if the lifespans in the wild are similar to what you found in the lab, or if they're different?
Surprisingly, yes. I would not expect that they would live similarly. I would expect in the lab, since we provide them like ad libitum food. But some studies show that it's kind of similar, like maybe on average a few days shorter in the wild, but that's it. So it's kind of representative of the wild lifespan.
Any more questions? One right there. Right here.
Hey, so I guess in the wild they're experiencing like quite different temperature. So in the lab, are you keeping them at like intermediate temperature, so they are not at the optimum?
Yeah, so we did the intermediate because the Swedish ones are colder, the French ones, so we picked one intermediate and thought that would do the job.
Thanks, Edim. Okay, our next speaker is Evi Savola from the University of Edinburgh.
Hi, so I'm Evie. I work over at the Walling Group over at the Institute of Evolutionary Biology at the University of Edinburgh. And today I'll be talking about whether the life history response to dietary restriction still persists with infection or injury. But just before I get started, just a huge thanks to my supervisors, people who helped with the project, and of course my funding bodies as well. Right, so dietary restriction, as the name suggests, is just restricting either the overall calories of food or a particular component in the diet. It's had a lot of interest over the years because it seems to be something that really consistently, as an environmental manipulation, extends both lifespan and delays aging. And it seems to occur across a wide range of species as well. So there's a lot of interest in this in terms of an aging society and seeing how we could possibly help humans with this. But even though it seems very evolutionarily conserved with a possible conserved mechanism, we don't actually really know why this happens, why it extends lifespan. I'll be talking about two different hypotheses here that have already been previously mentioned, but I'll just run through them quickly again. So first up is the hypothesis by Shanley and Kirkwood. It's sometimes called the adaptive resource reallocation hypothesis, and all they're saying is that because there's only a limited amount of resources, so an individual can either place them under survival or reproduction. So at times where there are these limited resources, it makes more sense to have them put into an increased lifespan, into somatic maintenance, and then there's an according lower reproduction. So at times of when there's fluctuating resources and resources come back, that's when there's reproduction available. The other hypothesis by Adler and Bondaryansky, they state that these limited resources are actually upregulating these nutrient recycling mechanisms with the sort of a goal that there's any sort of immediate reproduction, even in these very low nutrient environments. You could think about it in the way that it's an individual is kind of making the best of the limited resources, trying to use them as efficiently as possible. These nutrient recycling mechanisms, however, also might be limiting the individual in the way that it might leave them quite frail and quite unable to deal with any additional stresses. They could be helping with protecting against common laboratory causes of death, but at the same time, if you add any additional stressors, this would actually remove the increase in lifespan. So when we set out to look at this, we were looking at what would happen in these dietary restriction responses if we added in this extra stressor. So the two different models have quite different predictions of what would happen. So the Shanley and Kirkwood one would say that the normal dietary restriction response happens. So we always see an increase in lifespan with a delayed aging, and we would see a reduction in reproduction. Also, these responses would be the same in all environments. The Adler and Bondaryansky theory, however, would say that we would actually, with this additional stressor, we would see a decrease in lifespan with no delaying in aging, possibly some reproduction, but again, these responses would be different in these different environments. So this is what we set out to look at. Which one of these things do we see happen once we test this out? How do we do this? So in the lab, we used a population of fruit flies. It's a more heterogeneous genetic background that we have. And we took 600 of these females, individually housed them in these vials that have food at the bottom. And these different foods were different amounts of protein restriction that we were using. In fruit flies and in a lot of other species, it's been shown that it's actually the reduction in protein instead of a reduction in overall calories that's driving this increase in lifespan. On the second day of our experiment, we had these 3 different treatment groups. So the infection we use is Pseudomonas entomophila. It's a bacteria. And what we do is we take a needle and dip it into this bacterial broth, and then we stab the flies underneath their wings. So it's called septic infection. The injury group is the same, except there's no bacteria involved, and the control flies were just under CO2 anesthesia. our measures of lifespan or survival. We were looking at reproduction in terms of egg counts. We had some other aging measures as well, but I won't have time to go into those today, but I'm more than happy to talk about them later. Before I launch into what we actually saw, I'll be showing my survival data in terms of risk ratios, so I thought I would just quickly go through them. So essentially, the higher up we go on this scale, that means there's lower survival, and they're always being compared to this hypothetical individual at y=1. So it's really easy to make these direct comparisons of what happens when you're in these different groups. So then, for all these different proteins in the diet, we can see how they compare to each other. So what are the two different hypotheses predict we would see? So Shanley and Kirk would say that we see this very common diet restriction curve. So essentially, when we decrease protein, so we restrict protein, we would see this increased lifespan. This would only go up to a point, and then we would venture into the malnutrition stage. Adler and Pondriansky, however, would say that we would see the same pattern, but only in the control individuals. So again, same curve, but with injury or infection, so any stressor, this response would disappear, and we would probably get lower survival that might be slightly higher at these very high protein levels. Right, what did we actually see? So these are our Cox proportional hazards model predictions that we got. You can spot probably right away that the control and injury groups, so the green and orange points here, they show this very similar response curve. Interestingly, infection also shows this. It's just kind of lower survival overall. And I also wanted to highlight that the worst diets are over here, the very, very low protein diets. They did very poorly on them. But essentially, we saw the same patterns of these curved responses, so the predicted diet restriction lifespan increase. What happened to the other side of things, the reproduction? Here, I'm just showing lifetime eggs produced per female for these same diets. This time, the individuals are grouped to their treatment groups. With higher protein in the diet, we were seeing higher lifetime reproduction. It's a bit easier to pick apart the patterns if you look at the model predictions, which are here. And essentially, again, the curves look the same. They have the same patterns. Infection individuals are just slightly lower. So again, we're seeing these similar patterns where injury and infection isn't really doing anything drastically different. We can also look at the same data of how many eggs they were making, but we can look at it in terms of aging. So this next graph is showing these three different treatment groups side by side. And this time we have how many eggs they were making on the y-axis. And we can follow how many eggs the individuals were making across the experiment. These predictions can get quite complicated, so I just wanted to highlight how if we look at these diets that are very high in protein, so these very dark lines, they have a very much quicker decline, so these slopes are much higher. Essentially, they're losing their ability to make eggs quicker if you compare them to these lighter diets that are lower in protein. So once again, reduction in protein, diet restriction, we're seeing a delay in aging. There's some more complicated patterns in here that I won't get into, but the patterns across, even with injury and infection, aren't that much drastically different. We're still seeing the same delayed aging across the different infection treatments. So hopefully with that, I've been able to show that we still see these lifespan extension responses from diet restriction, even with these additional stresses of injury and infection. More specifically, we saw that lifespan was still extended, we still saw a reduction in reproduction, and we saw a delay in aging. So our results, at least the way we did these experiments, is showing more support for this resource reallocation theory. But I'd like to leave it there and take any questions.
Time for questions. There is one over there.
I was wondering about the quantity of the bacteria that you use for your infection, because given that Entomophila is pretty deadly for
Yes,
Drosophila,
yeah.
I was wondering what's the quantities you use when they die.
We used quite a low dose. So in terms of OD, it's like 0.005. And even that, with that amount, we were still getting quite a lot of deaths. I think we were just trying to aim somewhere that it's not low enough that it's not really doing anything, and not high enough that we were losing oral flies. So we were able to get these sort of very different rates of dying on these different diets. So I think we picked an okay one.
Any more questions? We have some time, so this is a good time to ask a question if you guys have. All right, let's thank Evie again. Okay, our next speaker is Moritz Lurich from ETH Zurich.
Thanks. Hi. Yeah, I'm Moritz, and I'll talk about work I did during my PhD, which I recently finished. So, as most of you, I'm interested in the factors that determine or drive phenotypic diversification, and luckily we already have quite a few really well-studied examples where we know the underlying ecological causes that define the selection landscapes, and basically the agents of selection responsible for the diversification. But there are also many more examples that we really know not much about, but that also show amazing variation in their phenotype. So just as in my study organism, which is a small aquatic crustacean, which you find in Europe and parts of Asia. And this is just a random sample from a deep lake in Switzerland. And you find this really at any place, there amazing variation in their pigmentation for a given body size. And previous work has shown, previous work from southern Sweden, where actually most of the pioneering work has been done for this study system. Previous work suggests that it's predation acting along a gradient of different backgrounds that is responsible for this phenotypic variation in pigmentation. So here is data I assembled from a previous field study, and it's a pooled data from 7 shallow lakes in southern Sweden, and the black dots and the gray dots and some of the white dots are individual isopods collected from different types of macrophyte beds that have a different background. And so the hypothesis is that really visual predation from fish predators or invertebrate predators or birds is responsible for these distinct morphotypic or pigmentation differences. This was a really nice observation, but there hasn't really ever been an experimental test of that. And so I went ahead for my PhD and in a factorial design manipulated the presence and absence of macrophytes and fish densities in these big outdoor mesocosms. And what I found was, to my surprise, that that pigmentation, size-corrected pigmentation, didn't really depend on the presence or absence of fish, but only on macrophytes. So isopods were much more pigmented in the presence of macrophytes than in the absence. And so I had to come up with an alternative hypothesis, and because isopods are detritivores that feed on macrophyte detritus, for example, and the other component is that isopods are born completely without pigmentation. So these are tiny isopods and they're almost transparent. So I thought, okay, developmental plasticity. So as we already heard a couple of times in this symposium today, is a form of plasticity where you can have the same genotype can have different developmental trajectories to a mature form. And in isopods, there's reason to believe that this is the case because we actually know quite a bit of the basis of how the biological basis of how this pigment is generated. So isopods biosynthesize tryptophan, which is an essential amino acid, into xanthomethine, which is an inert pigment that is present in the skin of isopods. And the interesting thing here is isopods really have to consume diet items that have this tryptophan because they can't synthesize it themselves. It's really only coming from macrophytes or microorganisms. So testing whether different amounts of protein with tryptophan affect the generation of pigmentation or the development of pigmentation. I conducted a lab experiment where I collected isopods from the wild, had them made in the lab, Then the females reared the eggs and gave birth to pale, small isopods that I then reared under 2 different protein contrasts, high and low protein concentration, with artificial pellets that I made with, balanced with yeast and starch to get to different protein levels, and then continued to rear them until they were mature in these microcosms. And it was a fairly big experiment, I thought, until I went to a couple of Daphnia talks today. But it was roughly 1,000 isopods coming from 29 families. I fed them ad libitum, did regular water changes, controlled temperature and light, and I took a picture of each isopod every 3 weeks, so it ran 12 weeks in total, which, based on life history data that is known, should give them time to reach maturity or develop towards an adult phenotype. And because this was a lot of phenotyping work, and I'm lazy, but I am also interested in programming, I wrote this computer vision package, which I also expanded into, it has more modules now for object tracking. There's a landmarking module, so if you're interested in this, come find me after the talk or give it a try. It's on GitHub or directly on the Python repository. Okay, so these are some of the results, and it's clearly visible that protein concentration, so here for growth on the right side, on the left side, didn't really have a big effect on how fast they grew, but as expected, the pigmentation rates, and this is a log scale, were dramatically different across all families. So it's safe to say that this is okay evidence for developmental plasticity in pigmentation in isopods. But more than that, I also found that protein concentration really strongly affects survival, which I was a bit surprised at first, because I thought, okay, high protein is good, is high quality, but there is, as we nicely saw in the talk before, there's an optimum level of protein in your diet, and it's shown in many study systems, mice, Drosophila, humans, that too much protein leads to increased mortality, and it's related to the breakdown of the proteins, which is costly. Okay, and then taking all this together, I wanted to test whether either developing pigmentation or body size faster or slower in the different environments affects your survival. And so I took all these traits, the rates, but actually, to make my life a little easier, only the linear part, and put it into one generalized additive model framework, and found this. So there's a lot going on here. It's on the x-axis. This is growth rate. This slipped away. And this is pigmentation rate, and the more yellow the background is, this is the actual model fit, the longer is the lifespan during the experiment. And we can already see that the two surfaces are different, and the thing I want to draw your attention to is the fact that here in the low protein, under the low protein diet, the range or the area of high fitness phenotypes is much bigger than under the high protein diet. So here is really only a small ridge, and some of them have a long lifespan or a longer lifespan. But here it doesn't really matter whether you're here or here. Under low protein concentration, anywhere within this peak region, you can have a longer lifespan. And there's a lot more to this, but in the interest of time, I'll stop here. But if you're interested, please come find me after the talk or later during the week. Okay, to sum up, so in the lab experiment, I found only small evidence for effect of protein on growth rates, but a really overwhelming evidence across all families on pigmentation rates. So higher protein that leads to increased pigmentation rates, which is support for my initial hypothesis. And then also, depending on which diet you grow up in, you have different options for, the amount of high fitness option is either limited, is constrained, or large, depending on the diet. And I also want to say that finding this strong evidence for plasticity doesn't exclude the Cripps hypothesis I talked about in the very beginning of the talk. But this is work in progress, and we're trying to find out more as we go. Thanks.
Any questions? Anybody? One over there.
Thanks for the interesting talk. So I'm wondering, so in your contour plot, because on the end you don't have so many data points, and do you think this will be an influence to what you see, the pattern you see here? Because if you, I don't know, if you shrink this graph and then change the color, maybe they will look different.
The surface is generated, it's of course an extrapolation from the model surface. But and if I were really strict, I would probably only show the areas that lies within the outermost points. But I think what's more interesting than the actual surface is the fact that both of them follow the same envelope of possible phenotypic, of possible developmental trajectories, but the different, there's a cost attached to these trajectories, and I don't know if I got your question right, but this is only the linear part of the slope where still a lot of isopods are alive, and then if they made it either to the fourth or fifth data point where they are either alive or dead, this then generates this contour plot. But
Okay, so you mean you already filtered the data before you do this plot?
No, I don't filter the data.
But you just pick the linear part of your...
I only take the linear part in the beginning, just to give it a little more power.
Okay.
And I still have a lot of individuals. But of course, it's debatable whether that's okay.
Yeah, okay, thank you.
Yeah.
Thanks, Moritz. Coffee.
Thanks for coming back to our symposium. And I have just a small announcement for the speakers of this symposium. I wanted to send you an email, but I don't know when it's going to be sent. But so tomorrow evening at 8, we have this dinner at Kokal and Roosevelt. And if you want to go together from city center to the dinner place, then we meet at city center near Aussi Bar. And it's there is a bridge in the Aussi Bar. And we meet at the bridge or the city center side of the bridge. You should be having this email at least tomorrow morning. And if you have any further questions, you can just email me about this dinner. But now I want to introduce Mario Bruining, and she will talk about the evolution of variance control.
All right, so good afternoon. So is this on? Yeah. All right, so this project is a collaboration between Princeton University and Radboud University, where I did my PhD research. So in all natural populations, we see phenotypic variation, where individuals differ in their size, color, and behavior, for example. So where does this phenotypic variation come from? So in the first place, it can be due to, of course, genetic variation. So individuals differing in their genetic makeup can express different phenotypes. And whenever these phenotypic differences lead to differences in fitness, we expect evolution by natural selection. Phenotypic plasticity can also lead to phenotypic variation in a population. And phenotypic plasticity involves a correlation between the phenotypic trait and the environment, where so-called reaction norms can differ among different genotypes. But there is a third mechanism that can also result in phenotypic variation, and that's the topic of today. So intragenotypic variability, where genetically identical individuals can still express different phenotypes, even when experiencing the same environmental conditions. And interestingly, different genotypes can vary in how much of this randomly phenotypic variation they express, due to, for instance, differences in their buffering abilities against stochastic, microenvironmental, or developmental processes. So, and this intragenotypic variability has for a long time been observed in artificial breeding, where farmers observed that, for example, variation in birth weight differs among different families. But recently, people also started to study this phenotypic variation in, for example, model systems such as fruit flies, but also in natural populations of, for instance, mammals and birds. And even more interesting, recently, we are now also able to detect genes that are involved in the regulation of this stochastic phenotypic variation. So a next logical question that comes up is this phenotypic variation, is this the outcome of, is this an adaptive response? So can we think of conditions that do select for this randomly generated phenotypic variation? So we wanted to address this in this study, and to do so we used a demographic population model. And here we looked at 3 different relationships between a phenotypic trait value and reproduction as a fitness component. And in each case, there's an interaction with the environment, so where the environment shapes the relationship. So in the first case, we looked at a linear relationship. So in here, the 2 different colors indicate 2 different environments, where you can see that the environment changes the slope of the relation. Second, we looked at an exponential relationship. And these 2 scenarios could reflect, for instance, relationships between body size and fitness, where you can imagine that in some environments it might be beneficial to be small, while in other environments it might be more beneficial to be large. And thirdly, we looked at an optimum relationship between a phenotypic trait value and reproduction. And this could, for instance, reflect the timing of germination in the year. So in some years, where there is a long winter, it might be more beneficial to germinate later in the season, while in other years, where when spring starts early, it might be better to germinate early in the season. And we also looked at different environmental conditions, so ranging from a fully stable environment to a strongly fluctuating environment. So in the modeling procedure, very briefly, was as follows. So each genotype or each strategy was characterized by two different values, a M value and a V value, that together determine the normal distribution where phenotypic traits of new offspring were sampled from. So the M determines the mean of the distribution, and the V determines the variance in the distribution. So larger V values result in a wider phenotypic trait distribution, so more random variation. So then for a certain M and V combination, we just done simulations through time, and given the environmental conditions, we calculated fitness every time step. And then we calculated long-term fitness as the geometric mean fitness. So then, when doing this for different combinations of mean trait values and variance in the trait values, we can obtain fitness landscapes as illustrated here, where you can depict the fitness for every combination. And I will now show you these results for the different scenarios that we assessed. So we have the different relationships between phenotypic traits and fitness, and we have the different environmental conditions. So now we'll first look at the linear relationship. So here in these fitness landscapes, the blue colors indicate a low fitness, and the red colors indicate a high fitness. So for these settings, you can see that there's clearly an effect of the mean trait, where in this case there's selection for higher trait values, but as you can see, there's no selection at all on the variance in the trait. And this is true for both the stable environment and for the fluctuating environment. So in other words, in this scenario, there's no selection favoring increased phenotypic variance, but at the same time, there's also no selection acting against it. It simply doesn't matter how much variance you have. So then let's look at this exponential relationship. So again, we see a strong effect of your mean trait, but now we also see that it's beneficial to have a higher phenotypic variance. And this is true for both the environmental conditions. So when there is an exponential relationship between your phenotypic trait and your fitness component, it's better to have a higher variance, and this is due to Jensen's inequality or nonlinear averaging. So now the third scenario. So this looks a bit more interesting. So now when we have the stable environment, it's best to have no variation at all. So you should just make sure that you express the optimal phenotype all the time. But when the environment is changing, now we see that there's selection for some intermediate phenotypic variance. And this is a bed hedging strategy. So let's look at this scenario a bit more. So what exactly determines where this optimum lies? So in this graph, I will show you in a minute the optimal variance for different combinations of environmental variance. So far we have looked at a fully stable environment and at a strongly fluctuating environment, but now we'll see what happens in between. And on the x-axis, we have the survival probability. So fitness, ultimately, of course, is the sum of how many individuals survive and how many new individuals are born every time step. And our phenotypic trait in our model affected reproduction. And so far, we have held survival constant. So for all phenotypic trait values and for all environments, we just use the same survival baseline. But what now happens if we start to vary this survival? So I will show you these effects on the x-axis. So this contour plot shows the optimal phenotypic variance for this optimum phenotype reproduction relationship as a function of how much the environment fluctuates through time and the survival probability. So again, we see that if there's no environmental variance, it's most beneficial to have no phenotypic variance, instead just express the optimal phenotype all the time. And we see that the higher the environmental variance gets, so the more strongly it will fluctuate, the more phenotypic variance will be favored. So now the effects of survival. So, and again, remember, survival is the same for all phenotypes and the same for all environments. So it really only changes the baseline fitness. But we still see that if you increase your survival, it becomes less beneficial to have a higher variance. Or in other words, to put it differently, if you have a low survival, it is more important to express phenotypic variance. And that is because fitness is calculated as the geometric mean fitness, so it's proportionally more affected by low values. So changing this baseline really influences what the optimal variance is. So summarizing, I think what we have shown here, at least from a theoretical perspective, we can certainly think of conditions in which it is beneficial to have increased phenotypic variance, even genetically identical individuals and even within the same environment. And that this is really a combination of the environmental conditions, so how much it changes through time, the demographic context, so the life history of a species, so what is the baseline fitness, and what are the other fitness components doing, and finally, very importantly, how your phenotype and your fitness, how this functional form looks like. And together, these factors determine how your fitness landscape looks like. So then finally, I would like to emphasize that, of course, whether or not we expect a population to evolve towards this optimum does not only depend on these fitness gradients, but also, of course, on the underlying genetic patterns and genetic variances and covariances. And in our paper that will soon be published, we also show how changing these genetic covariances can really change the evolutionary trajectories. So for instance, starting from a population here, by changing the genetic covariances between the mean and the variance, you can observe a very wide range of different pathways towards this optimum. So, and with this, I would like to thank you, and I'll be happy to take any questions.
This one over here.
So you mentioned that in the case of an exponential fitness function, the selection for higher variance was due to a geometric argument about the Jensen inequality, which is due to the curvature of the fitness function. So I was wondering whether in the case where you've got a Gaussian function, the curvature is changing depending on whether you're close to the optimum or far away from it. So is the result about the level of variance that is optimal there, depending on those change in local curvature of the fitness function as you fluctuate?
So thanks for this question. No, I don't think so, because in this optimum scenario, so it's really a different process behind this, because this is a bed hedging. So it's an increase in geometric mean fitness at the cost of a reduction in the absolute mean. So I don't think it has to do with the curvature of the function, but it does matter, of course, how narrow you make this fitness distribution. If you have a wider distribution, you expect less. So it's a combination of the distribution, the environmental variance, and how narrow the fitness distribution is.
Hello, everyone. I'm Yamur. I'm a PhD student at the University of Zurich, and I work on life history evolution under cancer risk. And today I'm going to present one of the models we work on with Hannah, which is on linking cell-level strategies to organismal life history traits. But before I start, I would like to have some acknowledgements. So I would like to thank Hannah for her input and her support, as well as URPP Evolution in Action and University of Zurich for funding, and Arizona Cancer Evolution Center for a lot of fruitful discussions, as well as the organizers of this symposium for bringing us together. Right. So organisms tend to start their life as a single cell, and they don't stay like that, so they have to get to some size. And they do this through some divisions and differentiations. And during these divisions, sometimes things go wrong, and DNA replication doesn't occur as it's supposed to happen, and some cells go bad. And these evil cells can divide uncontrollably and eventually can cause cancer and make an organism die. So the more of these division events you have, the more chances of mutations you also have. Then one would expect an organism like elephant that has a lot of cells and therefore needs a lot of cell divisions to have an off-the-charts cancer risk compared to something like mice. But however, when we look at the data, we don't see that. So this is in the x-axis mass times lifespan in logarithmic scale, and the y-axis you have the percent of necropsies with tumors from the zoo, and you see that it's kind of flat actually. And this is something called Piro's paradox. And one idea is that large animals might have better cancer suppression strategies to cope with this increased chance of getting mutations. There is some empirical evidence for this. So one is, for instance, they looked at DNA damage response in elephants versus in humans, and they saw that elephants had a higher rate of apoptosis when their cells were irradiated. But there are also other suggested mechanisms that can contribute to cancer suppression, such as telomere length and telomerase expression, as we heard about today in Pat Monaghan's great talk and rap. And there are some also other things like tissue structure and division and differentiation strategies. So these actually don't act alone. And our question in this model is how all of these strategies are optimized under different life histories. So for this, we use a model. And we have a cell-level strategy that is composed of 8 different traits. And these are all of these traits. I'm not going to go all over them, but there's things like, for instance, telomere length, but also how you respond to DNA damage, and there are things related to how you do your differentiation and division. And the organisms start their life as a single cell, and what they need to do is to get to a certain target body size, which is the target number of terminally differentiated cells. So these are the cells that contribute to somatic functions. And during these divisions, as I said, some mutations can happen and can cause cancer and kill the organism. So talking about death, death can happen due to different causes. So from a cell's perspective, it could be due to telomere loss, or it could be due to apoptosis as a response to DNA damage, but it could also be due to cellular extrinsic mortalities, such as like inflammation or just getting randomly killed. And from an organismal point of view, there are different causes of death, and this could be related to, again, extrinsic mortality, things like predation, but also some bad planning in terms of how many stem cells you have in the end, or just losing all your cells due to some sort of developmental failure, and of course, cancer. So this is the only equation I have, which is how we calculate fitness in this model, which is the proportion of tissue cells to body size. So we assume tissue cells are the ones that are contributing to any somatic kind of function. So all the fitness comes from how many tissue cells you have compared to your body size. And this is evaluated from the time you mature, the time that you get to your target body size, until you die for an organism. So we get this strategy, which is composed of 8 traits. And what we do is evaluate the fitness of this strategy across different extrinsic mortalities and across different body sizes, and optimize it using a genetic algorithm. So here are the results. And this results section, actually, we fixed 2 of the traits and just let 6 of them evolve. So it will always be these 2 fixed. And I'll just remind you what is fitness. So the first question is whether fitness depends on body size and extrinsic mortality. And again, in all of these graphs, you will have body size in the x-axis, and the y-axis will be fitness in this one. But yeah, so in the top you have the extrinsic mortality. So low extrinsic mortality here and high in here. And fitness decreases with body size. And you can see it more clearly when the extrinsic mortality is higher. And this is actually not super surprising. And actually, when you look at why this could be the case, you can see that, so here, I actually plotted it for bigger body sizes, where this gray shaded area is the area that you don't even have any fitness, so organisms don't even survive to maturation. And this is because getting mature takes longer as you get larger. And so this is in line with literature, actually, where larger body-sized organisms tend to have actually lower extrinsic mortality rate. And this is kind of supports the idea that it doesn't make sense to get larger if you are not going to even able to live very long. So then, just showing how different traits look like at the end of this optimization. So this is for 3 replicates, and here it's shown for the higher extrinsic mortality, and this is the fitness for 3 replicates. And actually, there is no clear pattern that arises with body size in any of these traits. So there is some pattern in terms of some threshold that is in some of the traits, but it doesn't depend on body size at all. But still, all of these 3 replicas have the same fitness in the end. So this kind of looks like there is an adaptive landscape of different cancer suppression mechanisms. And there are different peaks. The different combination of traits can actually lead to same sort of fitness peaks. But then I was curious, so all of these organisms of body size, different body sizes have evolved different combination of traits. What happens if we take the big strategy and use it for all the body sizes? And what happens if we take the small strategy and use it for all the body sizes? So here I will show that. So the gray one is the baseline, so the fitness they get if they use the strategy that is optimized for their own body size. And the blue one will be the big one, and the red one will be the small one. So if the big organisms or organisms of different sizes use the cell strategy that is evolved for small one, for instance. So what I was expecting was something like this. So the baseline to be like the highest, because that's the optimal strategy for that body size, and then the other 2 to fare worse than the baseline. So then I was kind of surprised when I saw the result. And so the small one actually is more or less how I expected. So it's sort of sucks for the larger body sizes. But actually, the big strategy works as good as the baseline across different body sizes. And this reminded me of one paper I read a while back, which is about the rate of mammal evolution, where they showed that getting larger takes 10 times longer than getting smaller. So getting smaller seems to be easier than getting larger, and this might be another reason why. So maybe cancer suppression, at least under these scenarios that we looked at, cancer suppression might be one of the things that getting larger requires some extra innovations, whereas getting smaller, you can still use the same strategy and be fine with that. So just to conclude, I've shown that similar outcome can be achieved, similar fitness can be achieved through a combination of different trait values, and this fitness landscape can be with multiple adaptive peaks. And actually looking at across tree of life, we see that there are many organisms that live long and or get very large, and these all start from a different point. So they might have, because of their evolutionary history, have evolved different combination of strategies that help them cope with the extra cancer risk. So this kind of emphasizes the value of doing comparative aging and comparative oncology studies to understand the mechanisms of cancer suppression. And then the follow-up questions are, then can a lineage that changes its body size, for instance, adapt to its new cancer risk? So we have a model looking into that, and the answer is sometimes no, but I'm not going to talk about it. But if you're interested, we can talk about it in a break about this model, which is on body size miniaturization from dinosaurs to birds. And then the next question, obviously, getting large is beneficial in, being large is beneficial in many, many contexts, but maybe cancer risk is one of the factors, like can it be one of the factors that constrain body size evolution? So with that, I'll just finish, and if you have any questions, I'll be happy to take them.
Plenty of time for questions. Just over there.
Thanks for this very nice talk. And I have a question about your only formula about the fitness definition. Can you tell me more about why you consider about these tissue cells like over the body cells, and what exactly is the tissue cells you considered?
Tissue cells are the terminally differentiated cells. So the others are the ones that still, like body is composed of tissue cells, and then which are the terminally differentiated cells, and the stem-like are stem cells who have the potential of dividing. So we just assume that organisms cannot become like a big blob without having this tissue or terminally differentiated cells to maintain their somatic functions. So that's how I look at the fitness. So those are two types of cells.
It's on the right.
So you said it doesn't make sense to become large if you cannot live long. And so this seems to be an assumption in your model. And I mean, there are at least many counterexamples. Agave are the largest species largest organisms in the asparagus family, or the giant octopuses, Semelparus. Agave, they reach this large size and they put a lot of into reproduction and die immediately afterwards. So I'm not sure about the validity of your assumption. I think it's more, the main benefit of being large is that you can produce a lot of offspring, and not that you can necessarily live long. That will depend on other trade-offs. No?
Yeah, maybe I have too much of a mammalian or vertebrate point of view when I talk about this, where it is. So the larger body size has a correlation with the average lifespan and extrinsic mortality. So what I was talking about this, I was thinking that it doesn't make sense to be an elephant if you are only going to live 1 year. But I was maybe thinking there's a correlation also with fast and slow continuum. So the larger organisms tend to be in the slower side of the continuum. But of course, there might be other, I haven't maybe thought this through. I don't know.
You indicated that each of these traits is contributing, and so there is no sort of single strategy. But are any of those traits more important or more tightly related to fitness, ultimately?
So, at least the 3 of them seem to have like a very strict threshold that if you go above that, it doesn't they don't go above some sort of level. But then I also try to fix them and see what happens, and then the rest of the things were like still all over the parameter space.
Hi everyone, I'm Janis, I'm postdoc currently in the University of Groningen, and I will speak about how hormone IGF-1 could be associated with vertebrate life histories in the wild. So animals have different strategies how they allocate their resources that have created diversity of phenotypes that we can observe. Since resources are coming from the same budget, it creates trade-offs, and those trade-offs are in part mediated by different hormones. I'm focused on hormone insulin-like growth factor 1, IGF-1, which is synthesized by growth hormone mainly in the liver, and when released into the bloodstream, it's carried around and bind with different IGF binding proteins, which could enhance, inhibit the IGF's functions, or binding proteins could have separate independent role in regulating IGF functions. Most of the knowledge still is coming from lab and from medical studies, however, that we have. About the main functions, IGF-1 has a wide range of functions. Increased insulin and IGF signaling is associated in reproduction. It enhances sex hormone synthesis and later on functionally interacts with them. It regulates bidirectionally with follicular selection and its development. It's also associated with faster prenatal development. It also is negatively associated with lifespan. Higher signaling is associated with decreased stress resistance and, increased mistakes in protein folding and increased prevalence in age-related diseases. It regulates growth. Higher signaling is associated with increased cell division, cell differentiation, and protein synthesis. And my overarching question was, does variation in IGF-1 levels explain variation in life histories in the wild? So, using passerine bird study system, we first did comparative study. We measured IGF-1 plasma levels and used to illustrate the species samples are collected from, in terms of body mass at least, it ranged from 5-gram goldcrest to a little over 500-gram hoodie grouse. And what we found that as a main effect, IGF-1 levels in plasma were not associated with the clutch eyes. However, when looked into as a continuous interaction, the IGF-1 levels were associated with clutch eye size negatively in species with longer parental care, which is associated with the slower pace of life. But it was positively associated with clutch eyes in species with shorter parental care. Maybe it's interesting to note that the pattern was exactly the opposite when looked at egg size. As a mean lifespan, IGF-1 was negatively associated with it. And negative association was even more pronounced in species with lower body mass, in species which are growing faster and have a faster pace of life generally. And interestingly, the negative association diminished when body mass of passerine birds increased. So our results indicate that the IGF-1 could be involved in trade-offs between reproduction and lifespan in its forms, and in having more numerous but smaller young versus having bigger young, but less in numbers. As far as we can tell right now, it's consistent with the lab studies and also with the mammalian study system outside the lab. But still, some experimental studies need to confirm in study systems which where habitat changes and you have to compete for resources. We did some experimental studies with regards to growth where we injected nestlings of pied flycatchers with IGF-1, and during the 13-day fledging period, in the end of this period, just prior to flying out of the nest, IGF-1 injected nestlings were approximately 15% larger than the control ones. And they also had higher plasma IGF-1 levels. And IGF-1 also showed distinct age-dependent profile. Chicks that are in the middle of their nesting phase, exponential growth phase, they had significantly higher IGF-1 levels during the period that they needed most. And IGF-1 levels dropped towards pre-fledging stage when they have reached the asymptotic body mass. So we know that IGF-1 is dependent on food, amount of food that you are getting, but we don't know really how it, in a time frame, how it happens. Therefore, we did a food supplementary study, and we showed that 1 hour after getting food, IGF-1 levels were already increased. But no such pattern was observed in the water supplemented group. And we did that study in jackdaw nestlings when they were 15 days old, therefore in the middle of their nesting phase. We also, that I'm not going to discuss here, did the brood size manipulation with gratids, where we, therefore, changing brood size, we indirectly change the amount of food they are getting. And it shows the longer change when the food supply is changed, and it showed exactly the similar results. When you have less young around you, you're getting more food per capita and you have higher IGF-1 levels. Therefore, if you are getting more food, your IGF-1 levels are higher and you grow more fast. But not talked here, but IGF-1 levels also, the investment, IGF-1 made investment into growth showed also the flexibility depending on the physical maturity and the condition you have achieved at that point already. But one thing that we still don't know is with what costs it comes like having, we know that having higher IGF-1 signaling and faster growth comes with upregulated antioxidant enzymes, and it also interacts with glucocorticoids, stress hormones. But it would be really interest of mine to see whether higher growth comes with a cost of in self-maintenance and survival in systems where you really have to work for food supply changes, and you have to compete for everything. And with that framework, I will leave you and take questions.
And do you have time for questions? Just on the left, or on the left.
Hi, so in your pied flycatcher study where you show that they are growing faster, so how do you think IGF-1 is increasing growth? Are they receiving more food? Are they receiving more food, or are they more efficient at metabolizing the food?
I think the latter, because we don't have extensive data on still whether we didn't approve manipulation there. Like 2 of them got IGF-1 and control injected within the same brood. We don't have extensive data on whether experiment had an impact on control, meaning that if you get the IGF-1 initial boost, you get bigger, therefore you have the impact over in competition. But at least other behavioral studies that we have done with the same system does not indicate that large nestlings within a brood have a kind of effect over smaller nestlings. I don't know this for sure. I have already planned to fill this gap in a follow-up study with those. Therefore, I can't give you the answer with confidence, but I don't believe so that it got more food and dominated over the controls.
Any more questions? If not, then thank you, Anis, and next we have Paul Debes.
Hello, I'm Paul, and I'd like to tell you why we think that maturation timing and body condition correlate genetically for a locus that's called VGL3 and Atlantic salmon par. Age at maturity is a central life history trait because it trades off probability for dying with reproductive success. And age at maturity or reproduction may depend on critical growth rates, but also energy stores and their depletion. In Atlantic salmon, we find one locus, the VGL3 locus, that explains 30 to 40% of age at maturity variation, and it has two alleles that we call the E, the early, and the L, the late maturation alleles. So VGL3 also associates with maturation, growth, and lipid storage in other species. Interestingly, this encodes a transcriptional cofactor that controls cell fate into either bone and cartilage lineages, so it supports somatic growth, but it also, or adipocytes, which would support energy storage. So one may ask whether VGL3 actually is controlling a resource partitioning between somatic growth and energy storage. We did an experiment to investigate the role of VGL3, and that is we created 41 VGL3 genotype half-zip families by crossing homozygous parents in a 2x2 factorial. Left are females, right are males, whatever you want it, to create homozygous and reciprocal heterozygous families. And we ended up, we replicated this a few times and ended up with a 3-generational pedigree for which we had also reconstructed the grandparents to avoid inbreeding when crossing the parents. These families were replicated to 2 temperature environments with a lifelong temperature difference of 2 degrees that we call warm and cold. And then we recorded length and condition at the end of the summer and maturity status at the end of the spawning time in winter. We found that indeed VGL3 is associated with a higher maturation rate, but we also found that male maturation probability associated with male length. And whenever we find an association between two traits, we should be asking ourselves, how is the causality? And there is, of course, the causality problem, and that is we don't know whether one trait affects the other, what is actually causing what. And once we're adding a GWAS SNP to it, like VGL3, we don't know where it actually starts and what affects what. Is it maybe a mediated association that we found? In Atlantic salmon, luckily, females do not mature during the first year, and this creates an opportunity to study traits that are unbiased by maturation. And we did this, so we did some multivariate modeling of sex-specific traits. To do the multivariate modeling, we first, of course, removed the length covariate for maturation probability and made it its own response. Now every trait will be shown for males and females separately, males black, females white, and of course nobody matured, no female matured. But we collect male and female length data and then assess whether VGL3 has an effect in both environments. And we could do the same for condition. And first thing we found was that all traits did not differ between, for the VGL3 effects, between the environments. So VGL3 is a very environmentally stable, has very environmentally stable effects. And the effects were consistently positive for maturation probability and condition, and they were also then significant, but for length they were negative, though non-significant. Thus, we definitely have VGL3 effects for male maturation and female condition. But to go back to the resource allocation, we found very similar proportional effects for condition and length in females, and that is they were antagonistic. So this would indeed support a resource partitioning of VGL3, and detecting length effects is a very challenging matter. Anyway, we also wanted then to confirm that the detected VGL3 effects are indeed genetic, and we did this by looking at the effect of including a VGL3 effect as the mean effect has on the pedigree-derived heritability. We would expect, if it's a true genetic effect, to reduce the heritability. And we indeed found, when we run a model with and without VGL3 effects, that it dramatically reduced the heritability for maturation probability of males, but also the between genetic environment correlation. And that is in agreement with an environmentally stable effect of VGL3. For length, we couldn't really find much difference in the heritability, even a slight increase. But when remembering that the effect, if there is one, was probably very small, we would have expected this. And the effect for condition was just like for maturation, a strong decrease in the heritability and also a decrease in the between environmental genetic correlation. Next, we asked whether VGL3 genetic effects on maturation have the same ranking as for condition. That is, if a male has a high probability to mature, is the female, the related female, does it have a high condition to? And we did this by looking again between models including and excluding VGL3, and did this between male maturation and female length, and male maturation and female condition, and found, when accounting for VGL3 effects, that between male maturation and female length, actually it increased the genetic correlation. This was a surprise because we didn't see any effects on the heritability, and I can't explain it. If anybody has a solution, I'd love to hear it later. But for between maturation and condition, we found what we were expecting, a decrease of the genetic correlation, which confirms that these two may be highly related, that maybe even condition mediates the effect that we detect for maturation. So when we now look at these effects in the worm, I prepared the last slide to show you these effects on the breeding values. And that is for between male maturation and female length. These are the predicted parental breeding values. All of the breeding values were, of course, many more. We see that when we remove the VGL3 effect, we get a significant correlation. And this is a bit puzzling for me, and I can't explain it. And I don't know if it's actually an artificially created relationship or not, by just excluding these effects when there's no effect. But for the relationship between male maturity and female condition, we found what we were expecting. We even found that these VGL3 effects match a positive relationship, and they may actually create this genetic correlation. So to summarize, VGL3 is a major effect locus not only for maturation but also for condition, and it has strong effects on the genetic covariance matrix between condition and maturation, and possibly also on length, but I'm not sure what it means there. And VGL3 may control the allocation between somatic growth versus energy reserves, but this may be very hard to detect. And what remains unknown in all of this is that when we think now that condition-mediated VGLE effects are what we're detecting when we're looking at maturation effects, this may not be entirely the case, because we don't know whether, after all, we're only looking at a genetic locus as a marker, whether there may be pleiotropic effects on condition and maturation, or we're actually looking at a mixed pattern where both of these mechanisms work at the same time. Thank you for listening, and there's a large study, so there a lot of people were involved.
Questions? There's one over there.
Hi, thanks for the interesting talk. So I'm wondering, when you mentioned that covariance, so this effect that contribute to the covariance between the condition and the maturation, right? So is there any other factors will also contribute to the variance, or?
Yes, definitely. There's a lot of remaining variance there. I mean, just the difference between the blue and the green, that's VGL3.
Okay.
And of course, there's a large error. But we see it in two environments, and for the heritability, we also have the between environment effect, or between sex effect, and so on. There's a lot more that I didn't mention.
Okay.
So directions of change are consistent at different levels, between sexes, between environments, between sexes in one environment and the other environment, and so on. So it's
yeah, because you also mentioned that there's a strong effect of this VGL3 on the covariance. So I'm wondering, relatively speaking, how much do you think this will be?
It's in the warm, it turns from 39 to close to 0, and from significant to non-significant. I think it's quite strong. For the length effect, I don't know what it actually means, because it turns significant when you remove VGL3, but this would have quite an effect, I think.
Okay.
So then it depends on allele frequencies, whether you have a significant or whether you have a genetic correlation or not.
Yeah.
This complete removal of VGL3 effect reflects having only heterozygotes. Of course, that's never going to happen, but...
Okay. Okay, thank you.
Okay, if there are no more questions, then thank you, Paul. Jonas.
Thank you for coming for the last talk of the day. My name is Shakira. I'm a PhD student at the University of Ljubljana in Slovenia. And I'm going to talk to you about body size in spiders, so I hope that you're not arachnophobic. And as we know, adult body size is one of the most important traits related to fitness. And the size variation that we observe in nature is a result of natural and sexual selection pressures. And in some cases, these selection pressures can differ greatly between the sexes. And that results in sexual size dimorphism. So spiders have one of the most extreme cases of sexual size dimorphism in terrestrial animals. But in this case, females are actually bigger than males. And recently, more attention has been paid to the role of developmental plasticity in the evolution of sexual size dimorphism. So in theory, the sexes differ between in developmental priorities that are mediated by sex-specific selection pressures. And the body size of females, this large body size, correlates nicely with fecundity. However, despite considerable research efforts, we're still unsure why are males so small. And the answer to this question seems to be very taxon-specific, which highlights the need to study the selection pressures at the microscale. So for this study, I'm going to focus on this species, Nephilingus cruentata from South Africa. And not only the males are much smaller than females, they also exhibit an extreme size variation. So you can see we have males that are below 2 milligrams and males that are over 12 milligrams. So for this study, what I'm interested is in the relative effects of genetics and two environmental factors on male adult size. And it's known that spiders are capable of adjusting their size and the time of maturation by adjusting the growth rates or adjusting the number of molts during development. And they do so as a response to changes in diet. And more recently, it was also shown that males are capable of adjusting their time of maturation in response to social cues, which is known as socially cued anticipated plasticity, also known as SCAD. And this is important because for males, maturing at a right time, that gives them an advantage in scramble competition. But more specifically, there's 3 out of 4 species of sexually dimorphic spiders where SCAD was found, so males matured earlier in response to female cues. However, this earlier maturation was not necessarily correlated to a smaller body size. So I'm also interested in the relationship between the time of maturation and body size. So for this study, what we did is that in a split brood design, we separated spiderlings from 14 females and 6 lineages. And they were split into 2 feeding treatments. Half were fed ad libitum, and the other half were fed 2 flies a week, so they were poorly fed. Once they reached the subadult stage, they were exposed to social cues, either male or female social cues. And once they reached maturation, we measured their body size and the mass, the developmental time, and also we looked at the developmental time or the length of the subadult stage. We had a very high mortality in the beginning of our experiment, especially the poorly fed males died more. And then when it comes to size, the poorly fed males matured at very small sizes, around half. And this is just as a reference. This is the distribution of weight in the parallel laboratory population. Now, when it comes to size, the well-fed males grew at bigger sizes than the poorly fed males, which is not surprising. However, the social cue treatment had no effect on size. And we can see that this response to the feeding treatment is consistent among the 6 tested lineages. Now, when it comes to the age at maturation, or the time during development, there was a significant effect of the feeding regime. And also, it correlated nicely with the size. But in this case, actually, early maturing males were larger in size. However, this relationship disappeared when you take into account the feeding regime. And there was a lot of variation. Now, when it comes to the duration of the subadult stage, which is when the social cues were presented, against our predictions, there were no significant differences in the length of the subadult stage in either well-fed or poorly fed individuals. And then we also took a subset of males, and we looked at the difference between the difference in size between the subadult stage and the adult stage, and there were no significant differences in the poorly fed group, but in the well-fed group, there was a marginally significant effect of the social cue. However, we have a small sample size for this, and I wouldn't consider this a true effect. This would have to be corroborated with a larger sample size. So what's left to answer is how much of the observed size variation can be attributed to additive genetic effects. So for that, we constructed an animal model using a pedigree of 3 generations in a full sibling design. And according to our model, the largest contribution to size variation were actually maternal effects. But because of our study design, we cannot separate the maternal effects versus the common environment of the clutch or the egg sac. And then our, the contribution of the genetics was quite low in comparison to other spiders and also other insects. So we had 16%, or in other words, the inheritance of size was 0.16. And additionally, when it comes to phenotypic plasticity, that was as a result from our experiment was 16%, and the residual variation was 18%. So in conclusion, in our case, there were no effects of the social cues on spiders. And what was interesting is that we have a low heritability and a high developmental plasticity. So males were capable of maturing at an extremely small size, which highlights the importance of looking at developmental plasticity in the evolution and maintenance of sexual size dimorphism, at least in spiders. And lastly, we found very strong effects of the maternal effects in the common environment that would be interesting to disentangle them in the future. And additionally, it would be interesting to look at if there is a sex-specific genetic architecture for size in spiders, in sexually dimorphic spiders, and or if perhaps they resolve this intralocus conflict by genomic imprinting. And I would like to thank my collaborators in the lab and my funding and the organization, and you for coming to the last talk. So thank you. If you have any questions, I'll take them now.
Any questions for Shakira? Two in the middle.
Hi.
No, you have to use the microphone.
Hi. Congratulations for your work. I want to ask you, there are some genus of spider, for example, Micratena, that have multiple species that have sexual size dimorphism, and some species that doesn't have this sexual size dimorphism. How is the panorama in the Nephila group? And also, do you think that what selections, maybe selective factors or selective pressures, are promoting this sexual dimorphism?
Okay, so there's two parts to this question. Sexual size dimorphism has evolved in spiders independently in all groups. And in the nephilids, all nephilids are sexually size dimorphic. They range from moderate to extreme. So even when the phylogeny was reconstructed, the ancestral state was already dimorphic. So then we would have to go back, but there's still some debate about where to classify nephilids and what outgroups to use, etc. And sorry, there was another part of the question. What other selection pressures? There's many hypotheses. There's sexual selection. It could be by female choice. There is also, actually, in these spiders, I was talking about scramble competition. But in many cases, males accumulate on female webs. So there could be also some male competition. And in that case, larger males are better fighters. So it would be, they would have a higher fitness if they were bigger and they were exposed to other males. So you have that, and then there's also differential mortality hypotheses. There's the gravity hypothesis. So smaller males require less energy and also are better at reaching females. There's many, many hypotheses. And, but there's no one single one that would be generalized for all SSD in spiders.
Hi.
Hi.
So when you measured the body size of the male, did you measure it at several developmental stages or several ages of the male? And do you always find a correlation, do you always find strong maternal effects across if you did measure at several time points?
No, I only measured the size of the males during the subadult stage, but only a subset of the data. And I did not make a model for this. Yeah. Thank you.
Thank you, Shakira.